Sound.
Producing organ found in birds. It is found to be present. It is a sound producing organ present in birds and found to be present lower to that of trachea. This is actually the seating. So I am giving you all these things. Okay. First I have to give you a chart and then I will be giving you all these things. Suppose sometimes question comes like getting what is marsupium? I think I discussed it. This class will wait a minute. This is a subclass. The Prototheria, egg-laying mammals having cloaca. Theria,
স্যার ওই সাইডেরটা আসছে না তো।
কি হয়েছে? Oh, sorry. They give birth to young one. So again there are two types. Infraclass. One is Metatheria, Eutheria. They give birth to tiny, immature offspring. And they give birth to So in case of Metatheria, they gave birth to tiny immature offspring, and these immature offsprings, these immature offsprings, they are to crawl to reach the marsupium abdominal pouch found in these animals. So these immature offsprings are to crawl to reach the marsupium. What is marsupium? Marsupium is an abdominal pouch within which the immature offspring of Metatherians will become mature. বুঝতে পেরেছিস?
Yes, sir.
So there is a term called marsupium. দেখো লিখে দাও তো বলছি আমি। Marsupium refers to an abdominal pouch found in female marsupials within which immature tiny offspring of gets matured. Clear? কোন জায়গাটা? First to an abdominal pouch which is developed in female marsupials, within which the tiny immature offspring gets matured. In kangaroo, marsupium is visible. A small, tiny offspring is found inside this marsupium. Look, this is the marsupium. The young one gets mature and trying to come out. Again, this is the marsupium. One young is found inside. This is the marsupium. Okay. Now, is it clear now? And Eutherians, actually Metatherians, example kangaroo. Dog, cat, human, etc. They are the Eutherians. So this is what I discussed. I think the Prototherians are the platypus, or you can go dark-billed platypus, and it is oviparous. As it is egg-laying, so it is oviparous. Remember this thing, oviparous. They are oviparous. So this is all about the subclass and infraclass under Mammalia. Clear? ঠিক আছে তো? Chapter is which chapter? Chapter numbers? Chapter কত রে এটা? সেলটা কত নম্বর চ্যাপ্টার?
8.
Very good. Cell and its functionality. Now, under this chapter, we need to go through cell, prokaryotic, eukaryotic cell, already discussed. I think you can remember that I already discussed the prokaryotic cell. And eukaryotic cell, you have discussed now. First of all, we have to just classify it like this. We have to discuss here plant cell and animal cell. These two. One is plant cell, other is the animal cell. In this heading, we have to discuss. Now, can anyone of you can give me just once difference, one main difference between prokaryotes and eukaryotes? Hello? In prokaryotes, yes. It is well developed, yes. Actually, in prokaryote, nucleus is primitive type. Nucleus is primitive type in prokaryote, but it is well developed in case of eukaryotes. Primitive type means actually what happens in the prokaryotic cell, nucleus actually nothing but a circular DNA. Here nuclear material is represented by circular DNA only. But here you will get a true nucleus. Actually, if it is a cell, just simply I am showing you. and this is the nucleate, and this is the primitive nucleus, and it is also called primitive nucleus. But in case of eukaryotic cell, so this is a true nucleus. Or well developed nucleus. Having four components, remember this thing. Having four components, and the components are nuclear membrane, nucleolus, chromatin reticulum, so a true nucleus has four components. The components are nuclear membrane, the nucleolus, the chromatin reticulum, and a nucleoplasm. So these four components are found, and then you can call it a true nucleus, or you can call it well developed nucleus. So now we are going to discuss all those cells where you will get this true nucleus. And obviously, other most important difference between prokaryotic and eukaryotic cell is membranous organelles are not found. Membranous organelles are not found. Membranous organelles are found. The other most important difference is membranous organelles are not found and membranous organelles are found. Membranous organelle means mitochondria, means suppose Golgi apparatus, means endoplasmic reticulum, lysosome, all are the membranous organelles. And in prokaryotes, membranous organelles are not found. You will get a membranous organelles like they are called ribosome. But membranous organelles are not at all found here. First thing. So this is the basic 2 point of difference I am showing you again. The devoid of true nucleus, and it is with true nucleus. There is here nuclear material is just DNA. Here it is actually made of four components: nuclear membrane, nucleolus, chromatin reticulum, and nucleoplasm. Membranous organelles are not found in prokaryotic cells like mitochondria, Golgi body. Here they are not found, but membranous organelles are found in eukaryotic cell like mitochondria found, Golgi body found. Everything is found. So this is what about the basic point of difference. Whatever found in a prokaryotic cell, already I discussed while discussing what? While discussing the kingdom, while discussing kingdom Monera. I think you can remember. I discussed everything, whatever found, cell membrane, what it is made of, cell wall, what it is made of, and even the nucleate, the plasmid, the different types of inorganic substances present, everything I discussed earlier. Now I am not going to, that's why it is written already discussed. Till I am giving you some specific point here only. But remember this thing, already it is. Now we have to concentrate on the eukaryotic cell.
Sir, prokaryotic cell নিচে লাইনটা একদম কি লিখেছো?
membranous organelles are not found in prokaryotic cells. এই হচ্ছে কথা রাখবে। But here membranous organelles are found in এর সবটাই মুছে দেবো একটু। I am showing you two types of cells here. So পবিত্র শুনতে পাচ্ছিস? And these are the lysosomes. Centriole. Then some small vacuoles may be found. So you see, cell 1, microcilia. Cell wall, cell membrane, then comes to this is the vacuole. Nucleus, rough ears, smooth ears. Mitochondria, smooth here. You'll get the Golgi body. Chloroplast. Centriole. Lysosome. Ribosome. You also get the ribosome. Plasmodesma. refers to the cytoplasmic connection between adjacent plant cells. Plasmodesma refers to the cytoplasmic connections between adjacent plant cells. So it's a plant cell and it is an animal cell. Oh, these are the microfilament. Sometimes small vacuole. So this is what about the plant cell and animal cell. Now you see the vacuole is quite larger in the animal cell, but it is smaller in case of the plant cell. Is it visible? Diagram দেখাচ্ছে? In plant cell you see cell wall is present. In animal cell, cell wall is not present. Cell membrane is the outermost boundary. In case of plant cell, you know, a large vacuole is present, but you know in animal cell vacuole is very too small. Then comes to that chloroplast is found to be present in the plant cell but not found in animal cell. Similarly, lysosome is found in animal cell, not found in the plant cell. Actually, microfilaments are found in animal cell, not in the plant cell. So in this way, you can differentiate between the plant cell and animal cell. So it's the most important diagram because if you can understand this diagram, it will be easier for you to understand the cells, plant cells and animal cells clearly. You know, mitochondria are there. The microvilli, microvilli. Microvilli. So we should have argued about these cells first before starting the cell. That's why I actually used to draw these two diagrams. What is plant cell and about what is the animal cell. Is it clear now?
Sir, animal cell এ Golgi body কোথায়?
Oh. Actually, Golgi body may be developed here also. Because where there is here, there is a scope of development of Golgi body. Where there is here, where there is endoplasmic reticulum, there Golgi body may be formed. কথা বুঝতে পেরেছিস? যেখানে ইয়ার থাকে সেখানে Golgi body ফর্মের চান্স রয়েছে আর কি। The product of the ear coming out to form the Golgi body. Remember this thing. The product of ear after being secreted will be forming the Golgi body. That's why Golgi body is not a permanent organelle. After that, from the actually terminal point of the Golgi body, this secretion is coming out in the form of vacuoles, and eventually Golgi body diminishes, and again a new Golgi body will be formed. কথা বুঝতে পেরেছো? So where there is ear, you will get the Golgi body. Clear?
স্যার
হ্যাঁ বল।
স্যার এনিম্যাল সেলে মাইক্রোভিলাই কি ওই উপরে একটু থাকে?
হ্যাঁ, মাইক্রোভিলাই উপরে মানে কি উপরে সাইডে থাকতে পারে। আমি একটা সাইডে দেখিয়েছি।
মানে স্যার এতটুকু জায়গাতেই থাকে আর কোথাও থাকে না?
Basically, you can get it here also. Basically, throughout the surface, you will not get the macrovilli. কথা বুঝতে পেরেছো?
হ্যাঁ স্যার।
Clear? এই দুটো একটা খুব important because from this you have to study all these organelles that are found here. This membrane you can call it লাগবে membranes of vacuole is called the tonoplast. Membrane covering the vacuole is called tonoplast. Membrane covering the vacuole is called tonoplast.
স্যার
হ্যাঁ বলো
স্যার টোনোপ্লাস্ট কি অ্যানিমেল সেলে থাকবে?
বুঝিনি tonoplast
ভেকিউলে কি থাকবে?
tonoplast actually membrane covering vacuole is tonoplast. Just a minute. Now we have to proceed towards the eukaryotic cell.
স্যার সি হবে।
হ্যাঁ কিছু efficiency of cell. Now it is solved as smaller cells are more efficient than larger cells. Smaller cells are more efficient than larger cells. Can you tell me why? Why it says smaller cells are more efficient than larger cells?
স্যার
Anyone?
এটিপি কম ইউটিলাইজ করে।
না না
স্যার
হ্যাঁ বলো বলো অতনু বলো লোয়ার সারফেস এরিয়া আচ্ছা আর কেউ
স্যার সেলের নিউক্লিয়াসে সব ইম্পর্টেন্ট প্রোটিন সেগুলো তাড়াতাড়ি পৌঁছায় কম ডিস্টেন্সের কারণে।
মানে বলছে না। আর কেউ
স্যার সারফেস এরিয়া ভলিউম রেশিওর কারণে
সারফেস এরিয়া ভলিউম রেশিও
কারণে
কি বুঝতে কিভাবে বলো এটা
স্যার সারফেস এরিয়া ভলিউম রেশিও এর জন্য
হ্যাঁ কি হবে স্মল সেলে কি বেশি হবে না কম হবে স্মলার সেলে
বেশি।
বেশি হবে।
this is the most important point. I think I discussed it in the beginning. Okay. First point, actually, the efficiency of cell depends on surface area volume ratio, nucleocytoplasmic ratio. So if surface area volume ratio is more, cell will be more efficient. If nucleocytoplasmic ratio is more, then the cell will be more efficient. Suppose I am showing you. three structures taken. Surface area 6 centimeter square, volume 1 centimeter cube, ratio 6/1. Surface area 6 into 2 square, 24 centimeter square, volume 8 centimeter cube, 3:1. Here surface area 6 square means 54 centimeter square, 27 centimeter cube, 2:1. So what is found? The cell volume. 1 centimeter cube means here surface area means surface area 6 centimeter square means here you see 6, actually in a cube there are 6 surfaces and the ratio is 6 as square, that's centimeter square, and volume 1 into 1 into 1, that's why 1 centimeter cube, ratio is 6:2. Now with the increase in the cell size, gradually surface area volume ratio decreases. So smaller cells have larger surface area volume ratio than larger cells. Smaller cells have larger surface area volume ratio than the larger cells. This is the most important thing. First, smaller cells have larger surface area volume ratio than the larger cells. So what is the benefit of surface area volume? Surface area volume ratio is more. There will be more exchange of materials. If surface area volume ratio is more, there will be more exchange of materials. And if there is more exchange of materials means the cell it becomes more active. Actually, active efficiency depends on the exchange of materials also. If there is more exchange of materials means that means the cell is more efficient. That's why it is said smaller cells are more efficient than the larger cells. Second thing, nucleocytoplasmic ratio. You see. Look, a cell's volume increases mainly due to the increase in the volume of cytoplasm. Because nuclear material almost not quite different, but the volume of cytoplasm increases mainly. So if the cell size increases means the volume of cytoplasm increases. That means you can say the volume of cytoplasm of a larger cell is much more than the volume of cytoplasm of a smaller cell. So whenever you say nucleus and volume of nucleus, volume of nucleus by volume of cytoplasm. So volume of nucleus by volume of cytoplasm, that is the value that we get is found more in this case. Volume of nucleus by volume of cytoplasm is more in case of smaller cell than that of the larger cells. That means nucleocytoplasmic ratio is more in smaller cell than larger cell. It is more in smaller cells than larger cells. So nucleocytoplasmic ratio is more in smaller cells than larger cells. So from this point we can say that what is the benefit? As nucleocytoplasmic ratio is more in the smaller cell, what is actually the benefit of it? The benefit is that the nucleus in a smaller cell can look after all the activities going on inside the cell much more efficiently. And that's why it is said if nucleocytoplasm ratio is more in a cell, his cell is said to be more efficient. দেখো তো বুঝতে পেরেছো কিনা? ডাউট থাকলে বলো বলো।
এই জায়গাটা কি বললে মানে নিউক্লিয়ার প্লাজমিক রেশিওর ব্যাপারে?
Like one thing, I just telling you one thing. Actually, a cell's volume increases due to the increase in the volume of cytoplasm mainly. Are you getting me?
Yes, sir.
If it is so, then if the cell size increases means if you get this ratio, volume of nucleus by volume of cytoplasm, and volume of cytoplasm is more, then you will get a less ratio in case of a larger cell. The value will be less. বুঝতে পেরেছো?
Yes, sir.
দেখো বুঝতে পেরেছো কিনা?
Yes, sir, বুঝেছি।
But it is more. So the nucleocytoplasmic ratio is more in smaller cell than the larger cell. বুঝতে পেরেছো?
Yes, sir. হ্যাঁ।
Now you see, nucleus is to control all activities going on inside the cell. Actually, all activities, all metabolic activities going on inside the cell are under the control of nucleus. So if nucleocytoplasmic ratio is more, then it will be easier for the nucleus to control all the activities efficiently. Then where the nucleocytoplasmic ratio is less, nucleocytoplasmic ratio is more, means it is more efficient. As in smaller cell, nucleocytoplasmic ratio is more than the larger cell. That's why smaller cells are found to be more efficient. So these are two basic points which determine efficiency of a particular cell. Clear?
Yes, sir.
Okay. Now we proceed towards the big cells. So मुझे देख तो मुझे with respect to plants and animals, the first component I am going to discuss is cell wall. Mainly made of cellulose, or some pectic compounds are there. Some other compounds are also there. Cellulose and pectic compounds is the outermost boundary of the plant cell. The outermost boundary of plant cell. So already I show you the cell wall. Now the question is, how is cell wall formed? How is cell wall formed? Remember these things. During the this is the telophase stage of cell division. In plant cell division. What is found here?
স্যার এটা কি টেলো?
হ্যাঁ বলো।
স্যার টেলো কি এটা ফেজ?
In cell division, the phase are prophase, metaphase, anaphase, and telophase. In cell division, the karyokinesis, cell division আছে একটু বলে দিই হ্যাঁ। Karyokinesis, cytokinesis. Division of nucleus,
স্যার
just wait. During cell division, হ্যাঁ কে তুমি কি বলছিলি বল।
স্যার
হ্যাঁ
স্যার যে টিলোফেজ আছে টিলোফেজে কি মানে প্লেট ফরমেশনে মানে সেল ওয়াল ফর্ম হয়?
হ্যাঁ দেখো আমি বলছি, ওয়েট করো একটু। During cell division, karyokinesis occurs first and it is followed by cytokinesis. During cell division, karyokinesis occurs first and it is followed by cytokinesis. And what happens here? In karyokinesis, nucleus undergoes division to form two total nuclei. I think you can remember that in prophase stage, nuclear membrane disappears. In the anaphase stage, metaphase stage, clear? Arranged in the equatorial region, the metaphase, the anaphase, division of centromere occurs. Daughter chromosomes are formed as they proceed towards their respective pole in the telophase. You see, they reach to their respective pole. And eventually nuclear membrane is developed to form two daughter nuclei. দেখতে পাচ্ছ? This is found in metaphase. Here you get the anaphase. Here in the telophase. Then in telophase stage, two daughter nuclei are formed. What is found in telophase stage? Two daughter nuclei are formed. This is what I am showing you here. At mid telophase, when two daughter nuclei are formed at two poles, then cytokinesis starts. What happens in cytokinesis? In cytokinesis, some granular structures are found to be deposited in the center, in between two daughter nuclei, and these granules are called these pectin granules are coming from So during plant cell division at telophase stage, in between newly formed two daughter nuclei, pectin granules are arranged in the pituitary region, and these granules are secreted from Golgi body. That's why sometimes question comes: why number of Golgi bodies increases in the plant cell during cytokinesis? Why number of Golgi bodies increases in plant cell during cytokinesis? So that they can help in the formation of prototor cells. Is it clear to you all? বুঝতে পেরেছ? Next, what happens next? After that, pectin granules undergo condensation to form a thin membranous structure called cell plate. Cell plate is formed. After that, cell plate undergoes physical and chemical changes to form middle lamella. Physical and chemical change to form middle lamella. After that, on both sides of this middle lamella now, cellular deposited to form the primary wall. So this Dr. Nucleus. So in a newly formed cell, actually cytokinesis is completed when primary wall is formed. With the formation of primary wall, cytokinesis is completed. Are you getting me? With the formation of primary wall, cytokinesis is complete. Now, gradually with the advancement of age, the metabolic waste products are started to be deposited enough to the primary wall, forming secondary and in sometimes tertiary wall. So initially when the cell wall is cytokinesis is completed, along with the primary wall, secondary wall is not going to be formed. Are you getting me? With the formation of primary wall during cytokinesis, it doesn't mean that immediately secondary wall is started to be formed. It takes some time. So I am showing you now the Medulla lamella is mainly made of calcium pectate. Actually, what happens? It is written here physical and chemical change. And during this physical and chemical change, when pectin granules further condense, this is actually under the physical change. And whenever these pectin granules combine or react with calcium ion present in the cytoplasm, forming calcium pectate, this is actually the chemical change. That's why medulla lamella is mainly made of calcium pectate. Primary wall is mainly made of cellulose and hemicellulose. Primary wall is mainly made of cellulose and hemicellulose. Is it clear up to this? What is primary cell wall made of? Clear? After, as I said earlier, whenever primary wall formation is completed in the young stage, immediately after this secondary wall formation will not take place. After maturity, after some time, when the cell becomes much more mature, then only you will get the secondary wall formation. এটা মুছে দেবো আমি।
হ্যাঁ স্যার।
স্যার প্রাইমারি সেল ওয়াল সেলুলোজের পরে এটা কি?
হেমিসেলুলোজ, হেমি HEMI, হেমি CELLULOSE। হয়েছে?
হ্যাঁ স্যার, ঠিক আছে স্যার।
মুছে দিচ্ছি, দেখ সব হয়েছে তো? Now you see, with maturity I am showing you now. This is the middle lamella. Inner to middle lamella. This is the primary wall. So remember for two adjacent plant cells, there is one common middle lamella. Because this is a cell, this is a cell. So for two adjacent plant cells, there is only one middle lamella. After that, secondary wall is formed. You see. Now, inner to the primary wall. Remember this thing. Inner to primary wall, secondary wall material is started to be formed. Now you can call it secondary wall. This one is primary wall. Middle level. So secondary wall materials are deposited inner to the primary wall. That means if the thickness of the secondary wall increases, then the cell is found to be dead sometimes. What is found in case of sclerenchyma cells in plant. Due to the continuous deposition of secondary wall, the amount of cytoplasm decreases, and so that ultimately the cell is found to become dead. All sclerenchyma cells are dead cells due to the evenly thickened cell wall, and such even thickness is developed due to the continuous deposition of the secondary wall, because it is found deposited to inner to the primary wall. কথা বুঝে গেছে? Because it is cell, এটা cell তো এখানে নিউক্লিয়াস, এটা যে কন্টিনিউয়াস ডিপোজিশন থাকে দেখা যাচ্ছে না। So if deposition continues like this, then there is no space for the cell to work. Then what will happen? Then the cell would become dead. কথা বুঝতে পেরেছিস? Materials, eventually the cell will become dead. Now, okay. Now we are not discussing this. Now regarding secondary wall, at least 3 layers. Not, you should remember this thing, it consists of at least 3 layers. That means more layers may be found. Three layers are S1, S2, S3. Sometimes you get S4, S5, S6 depending on the type of plants and the environment where this plant is growing on. Now if I am going to show you, suppose this is the middle lamella. Middle lamella. Then you will get the primary wall. Then you will get the secondary wall like this. So if I am going to level it now, this is a middle lamellar. Primary wall. S1, S2, S3. Secondary wall. So S1 is close to primary wall. That means inner to primary wall S1 is formed. Inner to S1 S2 is formed. And inner to S2 S3 is formed. Then S4, then S5, then S6. Maybe we may get a lot of layers under secondary wall. At least 3 layers to be there. The secondary wall is made of cellulose, heavy cellulose, then you will get the lignin, suberin, lutein, etc. So cellulose and hemicellulose are there. Beside this, you will get lignin, suberin, cutin. You will get the lignin, you will get the suberin and the cutin. Now what are the basic functions of these three? Whenever lignin is found to be deposited in the secondary wall, it is to make the cell wall hard. If there is huge deposition of lignin, it becomes so hard that it is found to become fragile, it is elasticity decreases. So with the deposition of lignin, the elasticity decreases. Primary wall is elastic as it is devoid of this such type of substances. So secondary wall is not so elastic because of deposition of lignin. If lignin is more, gradually is what? Elasticity decreases. But it is to make the plant organ very hard. Suberin is made of fatty substance actually, and it is to make the cells impermeable to water. Sometimes in some areas of the plant body, some cells are required to be made impermeable to water to actually protect the core area. Suppose you know, in case of root, so here the these are endodermal cells, below which pericycle, below which একটা মিনিট So from here, water enters here. But water is only allowed to enter the xylem. That's why cells of endodermis, which are found opposite to the xylem, are only thin walled. But other cells are required to become suberenized. বুঝতে পারছো কথাটা? একটু বড় দেখাই। So this cell is required to be normal, it's called passage cell. It is not, it is thin, not suberenized. But these cells are required to be suberenized, so that not allowing water to enter inside. That's why sometimes suberin is required to be deposited in the secondary wall to protect. Otherwise, what will happen? If all the cells are allowing water, then the core will be flooded then. কথা বুঝতে পারছো? বুঝতে পারছিস? So this is what about the role of secondary wall. You will get Q10 in leaf. Your Q10 is to check the rate of transpiration.
Sir, T cells to
Huh?
be.
Passage cell, passage.
Oh, acha, acha.
Allowing water to enter. এটা পরে বলবো আপনারা। So lignin make it hard, suberin make it impermeable to water, and role of Q10 is to check the rate of transpiration. বুঝতে পেরেছো? আজকে অবধি থাক। আমার নেক্সট দিন আমরা এবার সেল মেমব্রেন থেকে একটু ফাংশন বাকি আছে বলে সেল মেমব্রেন দিয়ে শুরু, ঠিক আছে?